Viewing these slides, and I'm not sure how much of it is me just being tired, but looking at them, there's a lot of stuff going on in electron transport. A lot of stuff that you probably won't ever see or need. Okay? So try and focus on kind of the larger issue within these. Where are these reactions happening? Where are your proton gradients forming, generating? What kind of power comes out of them? That kind of idea. So we'll be a little bit careful over analyzing the data put up on these. Okay? So big ones, recognizing where everything is happening. Okay, Krebs cycle is happening on the very inside. Okay, so we're getting a lot of our enzymes embedded in the membrane, or that internal or that inner membrane for your Krebs cycle. Our proton gradient in our electron transport is shuttling protons into the intermembrane space, so between the inner matrix and the outer membrane of the mitochondria. Right? And that's where your energy ends up forming. Okay? Here's a pretty nice summary, I think, overall picture of what's going on within it. I would probably make the argument that what you guys need to recognize is probably a little bit more like this. You have complex 1 that accepts NADH, okay, and shuttles its electrons to coenzyme Q. At the same time, it shuttles 4 hydrogens across the membrane. Okay. How does it do that? You don't really need to know that. Okay, so hence the big erase through there. CoQ will shuttle those electrons over to complex 3. Okay. There is a complex 2 also involved in this that's not shown on this, and that's because they can't show it nice and linearly, because complex 2 also feeds into complex 3. Okay. Complex 3 takes the electrons from CoQ. Okay, so NADH dumped electrons into CoQ. CoQ dumps those electrons into complex 3. Okay. Complex 3 shuttles those electrons around out to cytochrome C, okay, and as it shuttles those electrons out, we shuttle more hydrogens across the membrane. Okay. This enzyme is actually a pretty neat one, complex 3, and so I ended up finding a neat little picture on Wikipedia that we'll look at in a second that shows all the intricate pieces. Cytochrome C dumps its electrons into complex 4. Okay. Complex 4 shuttles those electrons down carefully enough that it can react with oxygen to produce water, and there's kind of our end respiration. At the same time, we again shuttle more H+ ions across the membrane. Okay, so a lot of H+ getting shifted out so that we can then use that to make ATP. We've generated a battery. At one side of our membrane, we have the positive, and the other side we have the negative. Okay, so we're just generating a massive battery here. It's then how do we take advantage of that charge. Another way to look at our summary, which and this one shows it a little bit better, because we can see the blue one is our complex 2. That's shuttling electrons to CoQ. CoQ shuttles it then into complex 3, which is in red here. Okay. So slightly different format of visualizing everything. You could go through and look at the reduction potentials, which I have seen some classes go through and do this, and I have no doubt that when I was taught this originally, we had to go through and look at cell potentials. Okay. Not a big deal for the sake of this class, but you could take each of the individual reactions that are happening within each of those complexes and look at their cell potentials, how likely they are to occur, and then stack and add those in the appropriate fashion to see how much energy is actually being released through a cell potential. Okay. Neat concept, not a huge issue for the sake of this class. I think there's probably a homework question that looks at it. Okay. And on a side note, so back to our reduction potentials, one of the things that we have to remember is when we looked at Gibbs free energy, a favorable or spontaneous Gibbs free energy was a negative, and yet when we move to cell potentials, it becomes positive. Okay. I have yet to look up the history on that, but I'm pretty sure that's just because these were two separate discoveries. Gibbs was looking at energy of all reactions, and then somebody else went through and looked at the energy in redox reactions to generate batteries and cell potentials. They didn't find out till later that those two things should talk to each other, because if we're looking at Gibbs free energy that looks at all reactions, a redox reaction should count. So how we monitor the energy in both systems should equate to each other somehow. And I'm going to guess that when Gibbs looked at it, he said negative was spontaneous, and that whoever went through and looked at cell potentials said positive makes more sense, because positive money, it makes me more wealthy, so I'm going to call things positive being a good thing. Okay? And then when those two fields decided to then talk to each other and communicate the relationships between them, they both said, F you, I'm not changing my approach. And so we ended up looking at a change in the sign. So that when you write out an equation to solve for them, if they both just agreed on the sign for a favorable reaction, you don't need the minus sign. Okay? Because they don't agree, I think that's why the minus sign really shows up. Okay? So just kind of be aware of that. Your sign can flip on you, depending on which you're looking at, cell potentials or Gibbs free energy. Moving to complex 1, NADH CoQ oxidoreductase is the name of our enzyme, or a potential name of the enzyme. Okay? NADH because it's using NADH, CoQ because it's using CoQ, and then it says oxidoreductase. Okay? Why oxido and reduct?
Using oxygen?
There's no oxygen at this stage. You're looking at NADH versus CoQ. Well, technically there's oxygen in CoQ. Okay? So there's an oxidation in one part and a reduction in the other part, and so they decided to put it all together into one big, nasty, massive name. Okay? So a lot of the enzymes we're going to be seeing in this have some kind of oxidoreductase name associated with it. Okay? Ultimately, your approach is shown kind of nicely in the summary that the textbook has provided. Your NADH shuttles electrons into FMN. FMN was a derivative of FAD. Okay, so instead of being the dinucleotide of FAD, it's a mononucleotide, and that's what they're showing over there. Okay, or what we've got on the far right. That then shuttles electrons into an iron-sulfur complex. That iron-sulfur complex shuttles electrons into CoQ. Okay, so we have various stages of stepping through the electron transfer, because we want to make sure that we can control the transfer of electrons at any given point. Okay. Next one, complex 2, succinate coenzyme Q oxidoreductase. It's another freaking fantastic name. Okay, what's happening in this case? We're taking succinate and we turn it into fumarate. Okay, and at the same time we produce FADH2. Okay, we've seen this before. There was an enzyme that did this. It was in the Krebs cycle, and we called it succinate dehydrogenase.
Yeah.
I think that's what it is. Okay. Why the hell do we have a new enzyme popping up with an entirely new name? As far as I can tell, it's coming back to the context of how each of these enzymes were discovered, because the enzyme is the same. The exact same. Okay. There is no difference. Okay. This complex 2 is the same enzyme used in Krebs cycle. Okay. It's going to come down to the context of where you're probably looking at the individual pieces. In Krebs cycle, we didn't evaluate CoQ, okay, or we didn't evaluate what happened to the FADH2. Okay, we just said, oh, it was produced and used somewhere on later down the line. In Krebs cycle, we were just concerned about taking succinate to fumarate. And so we said, that's our focus. So we end up looking at an enzyme name that does that reaction. Okay? If we look at what the rest of the enzyme does, to get FADH2 put into some place useful, it shuttles those electrons directly into coenzyme Q. Okay? So if we look at the other kind of product of our succinate dehydrogenase, it's making CoQ. It is complex 2. Okay, they're one and the same as far as their enzymes go. Okay. And that's ultimately how we're getting our energy too. We're shuttling those into CoQ. Complex 3, if you take a look at that, complex 3's got a massive amount of stuff going on within it. It looks like a giant, hairy, nasty beast. One thing to watch out for, or at least reference, is have some idea of what's going on with the Q. Q is in reference to quinone, okay? And if we take a look at our far right, we've got some changes that are happening to your quinone form. Okay, looking at the different oxidation states. Okay, depending on what's actually attached to the quinone, it gets slightly different names. This is referred to as a ubiquinone because your quinone is actually just the ring with the two oxygens. That's your quinone, whoops, with the double bonds. When you start attaching other things to it, it's now a quinone derivative, and they're calling it ubiquinone here. Any idea why ubi? Ubiquitous? It's used in a lot of places. Right? Really, really important for electron transport and generating energy. Okay? So it's in a lot of different places, and that's where our ubiquitous part of that name is coming from. So depending on the oxidation state, okay, you will look at slightly different forms of it. Notice we can start at CoQ and we can go all the way to QH2, okay, just like we did with our FAD. Okay. So what ends up happening is that when we hit complex 3, coenzyme Q will shuttle electrons, interestingly enough, into two parts of complex 3. Okay. It'll go into an iron-sulfur complex, which can later get shuttled out to cytochrome C. Okay. It will also react in something known as our Q cycle. Okay. So it reacts at a different point. So it's a little bit weird on what's happening within it. This figure, I think, does very little justice to what's happening within it. So let's take a look at what Wikipedia gives us. Let me turn on some lights here so we can take a look at this. I think this is actually kind of useful. So what we're looking at is our complex 2 and all the individual pieces within it. Okay? So our quinone binds, shuttles electrons, okay, to two different locations. And that goes fast. I can't talk through all that. And that's our first cycle. So quinone comes in, identifying the places within your protein. Your quinol, your reduced form, comes in, and it shuttles electrons to your iron-sulfur and to an intermediary at the bottom, which is interesting. We've got another CoQ at the bottom of this. It gets spit out. Another reduced form comes in, shuttles more electrons, which gets us our H+, and those electrons end up making it out to our cytochrome C. Okay. So for this protein to function, we're actually looking at two coenzyme Qs being involved in this. Okay. One part comes in reduced, one part comes in oxidized, and part of that reduction is actually going to reduce another coenzyme Q. Okay. A good question that might come out of that would be why? Why is it built in such a complex fashion? I have no idea. Okay? We just know that this is what's going on within it. Okay? So we're looking at a Q cycle because you end up consuming and producing coenzyme Q through that process. So kind of a neat little GIF showing all those individual pieces. Complex 4 takes cytochrome C, shuttles those electrons through the enzyme complex. Interestingly enough, we go through another copper center, or go through a copper center as opposed to an iron-sulfur, which eventually shuttles those electrons out to oxygen to produce water, and at the same time shuttle more protons across the membrane. Okay, those are kind of the basic details of what's happening within it. We can go back to our overall figure that shows all of that kind of sort of succinctly summarized. Again, NADH comes in, transfers to our FMN, iron-sulfur complex. That iron-sulfur complex in its excited state allows for the transfer of protons. Okay. The electrons can then get picked up by CoQ. CoQ then goes over, reacts with our complex 3 in 2 locations. Okay. Allows for the oxidation of cytochrome C, or sorry, reduction of cytochrome C. Regenerating CoQ, so CoQ is now in a decent concentration between both situations. Cytochrome C comes through and allows for the final reduction of oxygen. Okay. Kind of a neat little process, lots of individual electron transfer steps within that. Yeah. Questions about that?
What do you want us to do? All of that?
Yeah. So I'm less concerned about the details of what are all those individual pieces that are accepting electrons or within each of the complexes.
Like iron-sulfur, copper?
Exactly right. I don't care about those. What I'm concerned about are the primary groups. We start with complex 1, which accepts electrons from NADH, okay, which shuttles electrons to CoQ. CoQ shuttles those electrons to complex 3. Complex 3 shuttles electrons to cytochrome C. Cytochrome C shuttles electrons to complex 4. Complex 4 shuttles those electrons out to our oxygen. Okay. The next part to also associate with that is that within those steps, we also get proton transfer. Okay, we're generating a proton gradient. We're shuttling H plus across the membrane. One thing, or one reason why we might keep complex 2 out of this diagram, does it shuttle H plus? No, it doesn't. Okay. All it really does is shuttle electrons into CoQ, so that CoQ can then function. Okay. Properties of CoQ. Any ideas?
Like, what is it? Like, what is it? Like, what would its state be?
Well, we know it accepts electrons, so I mean we could go all the way back to where we actually see the structure of it. Okay, we're looking at CoQ on the far right there. Okay. So I'm not particularly concerned necessarily about the structure.
Oh, you mean like the reduced or oxidized state?
No, I'm not even concerned about that per se. Where is it located? Inside the membrane, which means it should be very, very hydrophobic. Okay, so if we go now all the way back, apologize for all the slide flipping. If we go back and look at CoQ, at its structure, what do you think? Hydrophobic, hydrophilic? Probably need to look up at the top where they show the full structure. It is relatively non-polar. Okay.
With that big old chain on there, right? Is that what?
That big old chain is a bunch of isoprenes, and it's kind of hard to see what that letter says. So it shows the parentheses there. Does it show N?
Yeah, it shows
That's what I was going to say. N means we've got some number of those, and it's usually a number that's fairly large. So we're probably looking at a minimum of 2 or 3, probably closer to 10. Is there anything polar in that? Nope, which would suggest very, very non-polar. It's going to stay pretty tightly bound within the membrane. What are you thinking, Jimmy?
So the ketone, the 2 ketone side chain or functional group, then they don't really affect it too much.
Are not going to have a huge contribution, primarily because of your isoprene units out at the far end. Okay. Yep. Okay. If we think back to organic, there's a general rule of thumb: 4 carbons outweighs 1 hydrogen bonding interaction. The ketones really have no hydrogen bonding. Okay.
Well, and maybe that
and you've
end
got
is closer to the, you know, because it's inside the membrane, it could be closer to the outside or the inside of the membrane.
True.
It does have a little bit of.
Yep. And it could, that part of it could kind of float through the membrane. And we'll look at the membrane structure a little bit later when we look at lipids. Okay. So CoQ is going to be relatively nonpolar, which means it should be pretty well confined within the membrane. What about cytochrome C? Okay, it's outside the membrane. We're looking at something that is going to be significantly more hydrophilic. Cytochrome C, while it is shown to kind of float over the membrane, is actually pretty tightly connected to the membrane. So it doesn't free float off into solution very well. It's going to be kind of almost like a gravity, if you will, held to the surface of your membrane. Okay. The next thing to consider also is the distances between each of these. Okay. When we draw them out, it looks like the distances are really, really big. Okay. We're not making any discussions about how big those distances are. Okay. Those could be exceptionally close to each other, or they could be really, really far from each other. To get it to happen efficiently, more than likely, they're pretty close. Okay?
I know you don't want to go into details too much, but the forming of water, is it really just have elemental oxygen coming in there and you have hydrogen?
So that's going to come in. I don't think I've got a slide for that. The elemental oxygen is likely going to bound to a metal center within your cytochrome. Okay? And that's going to be what facilitates your electron transfer. I'll see if I can't find an image that shows that structurally, but I'm fairly certain that you get that oxygen binding directly to a metal, and that polarizes the oxygen enough that when we shuttle electrons in, it can pick up hydrogens pretty quickly. Okay. There are other mechanisms where that shows up. So if we put iron, okay, the oxygen bound to the iron can share electrons with the iron, particularly if the iron has been, oxidized? Lost electrons, yeah. So if the iron is more positive, what happens? Well, then this oxygen can share electrons better down to it, which then means the oxygen at the outside better able to accept more electrons. Okay. If the iron had actually accepted electrons, okay, now what happens? Well, the iron could potentially share outwards, which then means these electrons could share outwards to potentially H+. Okay, kind of makes sense. So there's two different approaches to how we could go through and run that. And in those approaches, I think the second one makes more sense because the irons, your iron or copper, I think it was copper in cytochrome 4, are the ones that are accepting or gaining electrons through the course of the reaction. Okay. So more than likely, we shuttle electrons as a temporary storage onto our metal ion. That can then shuttle electrons up to our oxygen, which then starts to weaken the oxygen-oxygen bond. And yes, there are officially two.
You say
Okay. And so the end result out of that is you would end up kicking out your H2O, and then you would have, let's just call it metal, a metal oxide.
So is the oxygen is bound to iron, does that end up having a...
That then something else is going to need to have to come through and cleave that off, which could very well mean dumping more electrons onto your metal to shuttle them out. Okay. So I'll see what I can find, see if I can get a prettier image of what's going on with that than me scribbling. The next part of it is we now have this proton gradient with our big battery. We want to do something with it. Okay? And this is where ATP synthase comes in. ATP synthase is a big, large enzyme, okay, which is shown here. And what it goes through and does is provide a passive mechanism for H+ to come across the membrane. Okay? So in electron transport, it's active. We're using electrons to shuttle H+ across the membrane and thereby dropping the energy of our electrons, putting them into a proper oxidation or redox state to react with oxygen without exploding the cell. Okay? We've then used that energy to make this big old proton system on one side. Well, those protons don't want to be on a high concentration. They want to reduce, okay, or become more neutral. So to do that, they will have to then backflow across the membrane. The issue is that the membrane prevents them from moving through. Okay? So this is where ATP synthase comes in. ATP synthase provides a channel. Okay? So there's a little hole now on one side of our membrane where the ATP synthase sits in there, allowing for H plus to come into it. Okay? That channel is designed, okay, to now capture the H plus and say, you are more than welcome to come through my little slot here to get to the other side, but you have to move a proper direction. Okay? And by thereby moving around a sphere or around a cylinder to then make it to the out point of our membrane. Okay? By forcing it to then move around this, what we've now done is built effectively a dam and a power generator. We have a rotation happening. We can use that rotation to generate energy, much the same way our hydroelectric dam systems work at human scales. Okay? That causes a rotation to happen in this F1 system, which then forces ATP to be synthesized by bringing phosphate and ADP close enough to each other where they're now in the proper alignment to react. They just don't have the proper energy. And then that F1 system grabs them and forces them next to each other, forcing the bond to happen, okay, or forcing the bond to form. Dang it, I was going to look up a neat little video of this. Okay. Sorry, I just remembered that. So it's kind of a neat little process. We generate the proton gradient, and we allow passive protons to transfer back across, but we take advantage of that passive transfer through ATP synthase. Okay. Drugs can go through and uncouple this. Okay. So we could add drugs at varying stages through this process to prevent ATP from forming with this proton gradient. For instance, I could take a drug that goes through and it prevents ADP from binding. Well, if ADP doesn't bind, okay, I can't make ATP anymore, but I still have a hole in the membrane, which means protons can still move through and out. Okay. You'd just be wasting all of that energy. Okay. So depending on what we're trying to do with our drug, that may be useful, or that may be incredibly bad, okay, because you then shut down the ability to produce energy. Okay? So kind of a neat little system. If we take a closer look, so this is, of course, we're going to see more information about ATP synthase because it was one of my favorites. It gets broken down into two primary categories, the F1 and the FO. Okay. The F1 is in the cytosol, or not cytosol, in the matrix, okay. Not membrane associated. Okay, so we've got our, for lack of, let's just say not membrane. I can't come up with a better way. Our not membrane part is our F1, and then our FO is our membrane part. What things could we use to describe potentially the membrane part and the non-membrane part? The membrane part has a large percentage of hydrophobic amino acids within it because it needs to be in the membrane, which is largely hydrophobic. The amino acids up in the non-membrane part will probably be hydrophilic because that needs to associate out into the not membrane part.
So that's inside the mitochondria.
And this is all happening inside the mitochondria.
So the F1 is inside, not, so it's, you got the mitochondria and it's in the inside.
It's on the inside. And it's doubly inside. It's because it's not only inside the outer shell of your mitochondria, it's also inside that inner membrane. So there's the outer wall of our mitochondria, there's the inner membrane, and we have the inner membrane space, and we have the matrix. The F1, I'm fairly certain, is aimed at the matrix.
And so the FO goes through two membranes and inner space?
Nope.
Or just that
So,
inner membrane? Just the inner membrane,
Let's,
Go back a slide, it has it for you.
did they actually, oh, they did label it, yeah. If you check out at the top. Okay, the very top of that figure, the outer mitochondrial membrane,
Okay.
then you have the inner mitochondrial membrane, and that's where your H plus is, that inner membrane space. Okay, and then in the matrix is where you're generating that ATP. which is an interesting question. Where do we want ATP?
Well, we want it in there because we want to control it until we kick it out, because we don't want to.
We want ATP pretty much everywhere. So once we've produced it, it's now through two membranes on the inside of the cell. Eventually it needs to make its way back outside of all of that. Okay, so we're going to have to come up with some way to shuttle ATP outside of all of this and still use that energy. Okay. After we break down the FO and the F1, we then shift into all sorts of subunits that come within this. So when we're looking at ATP synthase, this is a good example of what type of protein structure. Quaternary structure. We have several different subunits that build within this. Okay? And they're all kind of listed there with their Greek letters representing them. You have alpha, beta, there's a gamma, a delta, epsilon, and then you have A, C, and B subunits. Okay, so they switch from our Greek prefixes to, or not Greek prefixes, Greek letters to Roman letters. I think that's right. Depending on which subunit you're in, be it the alpha or the F1 subunit or the FO subunit. Okay. B is kind of weird because B transverses two places. Okay. It connects both to the membrane-bound part and out into our matrix. If we take a look at the FO, which is what they're trying to show here, okay, here's our input hole. H plus can come up into it, and it can bind to the C subunits. Okay? And there's a whole bunch of C subunits all arranged in kind of a cylinder. And the way it is set up is that it cannot rotate, or can only rotate one direction. So the H plus can come in, but then for it to escape, it must actually force a rotation all the way around. Once it gets around to the other side, H plus can then funnel out the other end. Okay, so we only get a single direction rotation on our FO. Okay, which is important because if we didn't have that, what happens? H plus comes in and kicks real quickly backwards and goes right back out. We get virtually no rotation. Okay, well, very little rotation means very little energy produced. Okay.
Crazy, one more hydrogen causes that whole big old thing to spin.
Well, it's not just one hydrogen.
It's one thing.
Okay, it's a whole bunch of them trying to push across. And so one comes in and says, "Yay, I've made it across." Oh, no, I haven't. Okay, but now what happens to the structure when we bond to hydrogen?
It'll shift.
Why will it shift? What happened to the intermolecular forces when you bond to hydrogen? It changes, which is now going to cause it to go, oh, well, crap, potentially where I'm bound now is hydrophobic or hydrophilic, and I want to move a different direction. I want to avoid that like or that dissimilar charge, so it's going to shift. Well, now you expose a new slot, more H plus can come in. And again it goes, well, crap, there's a bad interaction here, I'm going to shift. Okay, and it can constantly keep that shift going as long as there's H plus to back it up. If there's no H plus there, then it can't come in. Same thing could theoretically happen from the other direction. H plus could come in from the top and cause a rotation the opposite direction. Okay, but the gradient isn't there to force the opposite rotation. Okay, which is an interesting question. I don't know if anybody's actually tried that. To switch the gradient to see if you can force the rotation to go the opposite direction. I would guess that they have, just to prove that something like that is happening. Okay, it'd be a neat experiment to test with or to play with. Okay, that rotation gets translated via the epsilon and gamma subunits up through the center stalk or the stator of your enzyme that then affects your alpha and beta subunits. Okay, so those start to rotate. Okay, your gamma and epsilon will rotate, and you can kind of envision them like maybe a stick coming off of that. Okay, as it rotates, that stick hits the alpha subunit. Well, now the alpha subunit has this thing poking in it, so what does it do? It moves, it changes its structure. By changing its structure, it now changes its affinity for ADP, ATP, or phosphate. Okay? But then the stick rotates again. Well, now alpha says, well, I can fill that space back. And again, its binding affinity for ATP, ADP, and phosphate changes, okay? Causing one of the next stages to happen. Okay? So when we go through and think about ATP, the things that are rotating, let's see. I think I can do it in red. Our rotations, our C subunit is rotating like crazy, okay, within our FO. Okay. I think it's, I'm not positive on this, but it sure looks like the epsilon is the only one that's going to rotate. Maybe, or sorry, the gamma. Maybe the epsilon rotates as well. Okay. But that's it. That's the only thing rotating. The alpha and beta subunits, the B subunit, and your A subunit do not rotate. Those are all locked into place. So what's happening is C just kind of rotates within that, causing some conformational changes within the rest of it. If alpha and beta weren't or were allowed to rotate, then you don't get any chemistry happening there because there's now nothing causing their conformation to change. They would just rotate with our gamma. So they need to be locked into place. That's why we have B, okay, holding on to it saying, okay, F1, don't move, but then B needs to be locked into the membrane. That's where A comes in. So you get this massive organization to lock everything into place, so the only thing rotating, okay, is through your center section. Okay? And then the rest of it just does conformational changes. Okay? So that when we then go through and look at it, say from, I'm not sure how you would reference, maybe as a top cycle, we've got varying stages happening within our alpha and beta subunits. Okay? They've gone through and tried to simplify that down into 3 potential binding sites, okay, which they call O, T, and L. Those 3 binding sites have varying affinities for ATP, ADP, and phosphate. Okay? So your open site has a low affinity for the substrate, okay, which we want a site to have a low affinity because you've got to let go of it. Okay? If you don't let go of it, that kind of sucks. We have a loose binding site. Okay? The loose binding site will bind ADP and phosphate. Okay? Those need to come in and interact. Okay? And that's when it's not catalytically active. Catalytically active. All that's really happening is we've now bonded ADP and phosphate. They're put into the enzyme so that we can now put the pieces together. Okay, imagine just taking 2 Lego bricks and holding on to them in each hand. Okay. Your tight binding site now brings those 2 pieces near enough to each other in the proper orientation that they are now forced to make a bond. You're taking 2 Lego bricks and you bring them into the proper orientation where you can snap them together. Okay. That's then our tight site. That's the catalytically active site. Okay. To exchange between each of these sites, our central stator is rotating and causing a conformational change that then allows for each of these changes in state. Okay? So our ADP binds. Okay? Once the ADP, yeah, they're showing ATP there anyway. Okay, so our ADP binds into the catalytic site. Okay? We get a rotation that causes ADP to then get much closer to phosphate. Another rotation causes ATP to form, and at the same time, any ATP that was left in the site gets kicked out. Okay? So you're getting all these little kind of steps happening within it. It's kind of a neat little process to go through and deal with. Okay. So there's kind of a nicer picture all by itself. What I was hoping to find, okay, is that, remember, during this whole process we have that stator, which is in the middle doing a constant rotation, causing each of those things to change conformations. We might say, well, how the hell can we possibly figure this out? Okay, there's a neat little YouTube video that someone was able to put together. They took a bunch of the C subunits. did they use C? I'm not, I think they used the C subunit, and they used the gamma and the epsilon. Whoops, that's not how you do epsilon. Close enough. Okay. But they got rid of the whole pretty much F1. Okay. And what they decided to attach at the end of this, okay, which not really drawn to scale, they attached a giant gold nanotube. Okay. Well, the reason they picked gold was gold is sensitive in our microscopy instruments. So what they could go through and do is they then filled the sample with a whole bunch of H+. And what happened? The H+ causes the C subunit to rotate. By causing it to rotate, the gamma and the epsilon rotate, which then causes the gold nanotube to rotate. And you can actually watch a video of seeing these weird sticks moving. Okay? And so it's absolutely fascinating they're able to do it. Add to this some kind of extra information here. If we draw this a little bit more to scale, we're looking at the C subunit, our other subunits, and then our gold nanorod is something like this. Okay? So we have a massive gold nanorod sitting on the end of this, and it is still being forced to move based off the power of this enzyme, okay, forcing that proton gradient. Within that, and this is kind of where the three states came about, is that they see when it rotates, it goes in roughly thirds. It does kind of third rotations, okay? And it now becomes questionable, well, why is it doing third rotations? Okay? If you took a rubber band and just held on to each end of it and then just kind of twisted gently on it, you aren't going to see much of a rotation until you start twisting enough that there's enough torsional strain that it forces it to rotate. So one of the potential theories here is that we're seeing these kind of three stages because we've put so much torsional strain on it that now it's forcing it to rotate. And we've released that stress, so then it stops. And then we crank it up, wind it up a whole bunch, and then it moves again. Okay? So knowing exactly what's happening within there becomes a little bit trickier, but these are pretty solid theories on how it's functioning.
Would that also, if it is doing every third, would that give time for each of the columns to change their shape without?
Each of those changes, so your time becomes a difficult concept to deal with here because you're looking at things happening on incredibly fast time scales. Okay, I think even for this rotation video, they actually have it running in slow-mo, and it's still happening like insanely fast. So it's hard to bring time into that argument, okay, at least at this stage. So super neat molecule. I would highly encourage you to go out and read about it some more because it's kind of fun. Last two things, which at least a few of you started commenting on in the last section, and I hadn't commented on it yet. We can look at the phosphate or phosphorylation to oxidation ratio to see how much ATP is produced for oxygen being oxidized. Okay? And we'll notice that when we look at our phosphorylation to oxidation ratio, NADH produces more ATP per molecule than FADH2. Okay? And it just has to go with how the electrons are getting transferred and how NADH and FADH2 are making it into the electron transport chain. Okay? So NADH is a much more efficient transfer of energy. FADH2 happens to store a bunch of that or not allow for the transfer of that quite as easily, which gets us to our shuttle mechanisms. Depending on how NADH and ultimately, and potentially FADH2 get to our electron transport chain, we're going to get different energies produced. Okay? And so there are two primary mechanisms for getting that NADH across. That's your Glycerol-Phosphate Shuttle or your malate aspartate shuttle. Okay? And it just so happens that we get slightly different efficiencies of that NADH transfer. Because we get a better NADH transfer, we get more ATP produced. Okay? So that's what they're trying to go through and show with these next sequences. If we go through and look at where glycolysis is happening, glycolysis is outside the membrane. How do we get those pieces, or outside the mitochondria, how do we get those pieces into the mitochondria to react? Okay. Well, it's not a passive transport, it's an active transport. So the process of getting those things in produces varying amounts of energy. Okay. And that's ultimately our big difference between the two shuttle mechanisms, which then means we have two different types of, or different ways that we could produce ATP. So depending on which mechanism you're preferring to keep your focus on, you're going to end up referencing different amounts of ATP being produced, okay, depending on each cycle. And that goes back to just plain old cellular respiration. When we had talked about this sort of previously, what I had been referencing was the efficiency of ATP, or ATP synthase. Depending on the organism, there are varying amounts of H plus to generate ATP, a single ATP, okay? And varying C subunits. So less C subunits means less rotation, which could mean potentially more or less efficiency depending on how much rotation you need to force ATP to happen. So a lot of the numbers become kind of variable depending on the mechanism you're looking at for getting stuff from your glycolysis into your mitochondria, and it also depends on the efficiency of the organism doing your ATP synthase. Okay. Your big summary, for those of you doing the MCAT, sorry, you should probably know all of that. Okay. Yeah. I'm glad. I think that was probably when I was like, I don't think I want to do the MCAT. That said, the ones that I've looked at, the MCAT versions I've looked at, don't really ask many questions about cellular respiration, at least the practice things I've seen, which I thought was kind of bizarre. Okay. But yeah, you're kind of responsible for knowing a lot of those numbers. Okay. Which we've already walked through and done. I mean, ultimately, it's just a question of how you stack and find those values. Okay. Questions about any of that? Hooray, respiration. Oh, so nucleotides. I did put them earlier. Good. Okay. So these are our main things. So within Chapter 9, we have the nucleotide introduction. So this is kind of looking at our bulk pieces on how we build our genetic information. Okay. And again, this is a section where if in the process of writing something up, you've been like, I was going to talk about that, then just let me know and I'll stop talking about it. I already tried to delete things that I felt should have been addressed in your reports and your presentations already, so hopefully it'll be relatively minimal. So, first thing we got to look at are nucleobases. Okay? The bases are categorized into two main sections. You have your pyrimidines and your purines. Okay? However you want to remember that is entirely up to you. For some reason, what stuck with me is they're kind of opposite of the length of their name. Pyridines are 1 ring, and purines are 2 rings. So your purines tend to be larger than your pyrimidines. Okay. You've got your basic structure shown within your purine and pyrimidines on the far left. Slight modifications give you the different nucleobases. Okay. Within your purines, you only have adenine and guanine. Within your pyrimidines, you also have uracil, an extra type of nucleobase. That is only found in RNA, not in DNA. Okay. We then move up to the nucleoside. Okay, well, what changed when we moved to the nucleoside from the nucleobase? We added a sugar, that's it. Okay. The most common sugar used for our nucleosides are beta-D ribose, or our beta-D deoxyribose. Okay. The only reason why I would then address this next part is because on a test where I shouldn't have gotten this question wrong, I did. What does deoxy mean? What does D mean? It's removal of oxygen, that's it. So when we look at this structure comparison between ribose and deoxyribose, what happened? You just lost an OH. That's it. We removed one OH. Everything else is exactly the same. Okay?
It's just an O, right, because you still have the H.
It's a trade of an oxygen for a hydrogen on that structure. Sure. Okay. Why might we want to do something like that? Where do we find deoxyribose? In DNA. In DNA, the D stands for deoxy. Again, one of those reasons why I was like dumb. I shouldn't have missed that question. Okay. So we got rid of the oxygen. So why do we want DNA to not have that oxygen? Oxygen's electronegative, which means oxygen makes the molecule more polar, more reactive. What do we want our genetic material to be? Stable. We don't want it to be reactive. The more reactive it is, the more likely it reacts and changes, in which case our genetic information leads to mutations, which could be great in a very variable environment. We aren't in a very variable environment. We're trying to maintain a standard state or a steady state. Okay? So deoxyribose is used in DNA as kind of a stabilizing factor. Okay? It limits the potential reactivity of that nucleoside. Okay? If you want to deal with numbering on where your ribose is attached, you'll notice that the nucleobase is attached to what position on the ribose? Position 1, which is the easy answer. Now give me the harder answer. It is the beta is in reference to where it's located. Yeah, but there's something else. That's the anomeric carbon. Okay. So we're bonding our nucleobase to the anomeric carbon of our carbohydrate. Okay. The next part is dealing with numbering on the nucleobases. Okay. In your pyrimidines, it's bound to nitrogen number 1. In your purines, it's bound to nitrogen number 9. How do you know to number it that way? Whenever you find out, please let me know. Sorry, that's all I got. Okay. Nucleotides. So we started with nucleobase up to nucleoside, now up to nucleotide. What's changed? Backbone means we've attached the nucleotide to another nucleotide. There's no, all it is we've added a phosphate. That's it. Okay. So our nucleotide now has the phosphate, which then means we now have the ability to link it to other or other nucleotides to form DNA or RNA. Okay. So our nucleotide is now the combination of all three things: the nucleobase, the or our sugar, and our phosphate. And we now have the nucleotide. Okay. And poking around at some standardized tests, I have seen questions about knowing the difference between those. Okay. So kind of watch out for that. Just be aware of it. There isn't a whole lot else to reference. When we go through and look at those, the RNA or our ribonucleic acid becomes a little bit trickier to evaluate because there's now 2 potential alcohols that could react on the ribose. We'd have position 2 and position 3. Okay? If you think back to maybe your biology classes, when you look at DNA synthesis, it always happens in which direction? Okay. So you guys just gave me the 2 numbering systems, which is fine, 5' and versus 3'. Okay. When you're talking about transcription or it's replication or transcription, we will need to reference which direction is correct. It's always fun to admit this. It's just a memorization thing, and I've forgotten it. I think it's 5' to 3'. What is the 5' and the 3' in reference to?
The orientation of the nucleotides.
Okay. Our numbering starts up on the nucleobase as our 1, 2, 3, 4, 5. Okay. Why do we then reference our carbohydrate as 1', 2', 3'? Well, if we didn't include the prime, we could confuse the numbering on our carbohydrate with the numbering on the nucleobase. Okay. So we end up including the prime to now reference the positions on the carbohydrate that are reacting. Okay. And when we go through and look at that, we've got the 3' position, and we have the 5', the number greater than 4' position. Okay. Those are now our two reactive sites. Those are the primary ones for reactivity, particularly when looking at DNA, because in DNA there is no 2' alcohol. Okay. In RNA, we do have the potential for modification at that position, right, which adds now variability, which could potentially be a good thing or a bad thing, depending on what we were trying to do. Okay. So our 5' and 3' are referencing those two locations and how those locations can connect to other nucleotides. Okay? Kind of makes sense? Okay. And actually, one more.
So when they connect, the phosphate from the 5' connects to the 3' of another one?
Yep, to the 3' oxygen of another one. Exactly right. So we'll kind of show this linkage. We're connecting the 3' of that top one to our 5', whoops. And actually I'm going to do it this way. To our 5' position of our other one. So somehow those are linked. Okay, here's why I didn't go super creative there. We want to know what is acting as the nucleophile, what is acting as the electrophile. What links to what?
And the oxygen?
So an oxygen-oxygen bond would be a peroxide bond, and those are highly, highly reactive. Think about hydrogen peroxide. Okay, you're really told to not drink that because that would kind of kill you. Okay, because it's super, super reactive. So we don't form an oxygen-oxygen bond.
Alcohol is acting as a nucleophile on the phosphate, the P, and then one of the oxygens on the phosphate, then we get some hydrogens, we kick off as
Our oxygen will act as the nucleophile. The phosphorus on our 5' end will act as the electrophile. The bond we're forming, whoops, wrong eraser, is between the oxygen and the phosphorus, and then we will eject one of those other oxygens out to form H2O. Kind of make sense? Okay. When looking at whatever it is, translation, transcription, and replication, that would be something I would suggest you kind of hover around, which I think is all you, Jimmy. I think you're in that area, or your transcription.
Mine is replication.
Your replication, your trans. I always
Which
get.
word it is?
I can't either. I think it's transcription.
What is the first part? Whatever that is. I think transcription.
You're looking at transcription. Okay. You should kind of address that same concept just to make sure. Just be like, hey, we already talked about. Remember those linkages. Okay. But yeah, so those are your nucleotides. Ta-da! Nucleotides. DNA primary structure. Okay, and this is where I'm going to try and be careful and not cover yours,
Hey,
Diana.
so just review, so it's okay.
In our primary structure, we reference our nucleotides linking through our backbone and our 5', 3' kind of connection. if you are going to reference your carbohydrate, you may see a D showing up in your linkages. So if we tried to reference the primary structure for that strand, we could go through and say, well, this is thiamine with a T, guanine with a G, cytosine, and adenine. If we wanted to further emphasize that it was a deoxy system, we could just draw in D. Because otherwise, it could have been RNA.
Well,
Okay.
no, 'cause it has T in there, and you wouldn't have a T.
Well,
like you.
Fine, fine, deal with that. Okay. So you might see that D getting dropped in there. That's to help reference that you're looking at DNA, not RNA, in your primary sequence. Okay. Secondary, tertiary, and quaternary structure. So at this point, this is just a single strand of DNA. Okay. What happens when that DNA interacts with other molecules? Are we going to get secondary structure? Yes, and that I'm
The dummy hitter.
leaving all on you and a little bit on you, but a little bit less so on the secondary structure. Tertiary and quaternary structure, I doubt anybody's going to look at. If nobody looks at it, I'll try and kind of back talk about it just a little bit. Okay. But we'll have to end right there. And then after that, I get into quaternary structure,